Hello and welcome back student to your video sessions. I am Shruti Banerjee from Holy Hearts Educational Academy, Civil Lines, Raipur. In our previous session, we have discussed about the cellular components. In those cellular components, we studied about endoplasmic reticulum, lysosome and ribosomes. We also studied about the cytoplasm. In continuation with that topic, today we will cover more important again cellular components or the cell organelles, right? So for today it is Golgi bodies. Now if you see this structure properly, you will see that there are curved tube-like structures which are again membrane-bound structures, because we are studying about the eukaryotic cell, and we know that membrane-bound cell organelles are absent in prokaryotes and are present only and only in eukaryotes. So in eukaryotes we see Golgi bodies, which are again tube-like structures, slightly curved, and they are arranged parallelly over each other, right? There is parallelly arranged structures. From these tubes comes out some vesicles. These vesicles helps in transporting. Okay. Now what is the function of this Golgi body? Golgi body is known as the packaging house of the cell. What do you mean by the packaging house? It will collect the substances made by the ribosome or the channelized by the endoplasmic reticulum, and it will pack them inside these vesicles. Just like you order something for delivery, the material which you have ordered always comes packed in a bubble wrap or in a cardboard structures. Similarly, the materials like enzymes, proteins or the fats which are synthesized by other cell organelles needs to be packed before they are transported to the target sites of the cell. And which organelle helps them? It is the Golgi bodies, right? So whatever materials are synthesized by the endoplasmic reticulum are packed and dispatched by the Golgi bodies. Now if you see what happens, like let's say ribosome has produced certain kind of protein. Those proteins will be channelized by the endoplasmic reticulum and it will come inside the Golgi bodies. Golgi bodies will take those enzymes and pack them in the form of vesicles. Can you see newly formed vesicles? These vesicles will bud out and go to the target site. So apart from packaging, it also helps in the formation of lysosomes. Now you have studied in your previous session that lysosomes are the suicidal bags of the cell, which helps in killing the foreign bodies and in some case also helps in killing its own cell. That is why they are suicidal bags. So they require powerful enzymes, hydrolytic enzymes, and those enzymes are given to them by again Golgi bodies. In many cases, Golgi bodies have also been found to make complex sugars from the simple sugars. So if there are simple sugars present inside the Golgi bodies, they can pack them and convert those simple sugars into complex sugar and easily transport them to the target sites. So this was Golgi body. The next important organelle which we will be studying is mitochondria. Now mitochondria is one of the most important cell organelle, as it is the powerhouse of the cell. A cell cannot last without a mitochondria. Why? Because it produces energy, and this energy is produced in the form of ATP. You can ask, what is energy for the cell? Well, for the cell the energy is ATP, adenosine triphosphate. These ATPs are known as energy currency. Like suppose a ribosome wants to produce protein. For producing, the factory needs fuel, and that fuel is in the form of energy. And from whom it will take the energy? It will ask mitochondria, please give me ATP so that I can produce protein. Golgi body wants to transport it packaged material, those vesicles. Again, to transport it requires energy. So from whom it will ask? It will ask mitochondria, give me ATP. That is why ATP is known as the energy currency. If you see the structure of mitochondria, you see that it is again membrane bound structure, but apart from the other structures which we have seen, mitochondria is made up of two membranes. So mitochondria, remember, is a double membrane structure, outer membrane and inner membrane. Inner membranes are folded in a specific structures, and these are known as cristae. Cristae are the folded finger-like projections of inner membrane of mitochondria. These membrane foldings help in producing ATP. Now inside the inner membrane, this membrane again is present the mitochondrial matrix. Mitochondrial matrix is nothing but again a jelly-like substance present inside it, and it contains its own DNA, mitochondrial DNA. You should know that mitochondria is one of those cell organelles which are autonomous in nature. Now what do you mean by autonomous? Autonomous structure means those structures which have their own ribosome and DNA. Whatever organelle today previously we have seen, like lysosome, ribosomes, or the Golgi apparatus previously which we saw, they do not have their own DNA. They depend upon the cell's DNA to carry on their function. But mitochondria does not depend upon the DNA of the cell completely. It has its own DNA. That is why mitochondria is known as semi-autonomous structure. Okay. Next organelle that we will be studying are the plastids or the chloroplast. Now plastid or chloroplast I have written, but actually chloroplast is a type of plastid. Now what are these plastids? First understand these plastids are present exclusively only and only in plants. Animal cells do not have plastids, right? And these plastids are, although they are of 8 different types, the 3 types which you need to know now are chloroplast, chromoplast and leucoplast. So plastids are the kitchen house of the cell. Okay, they are the kitchen of the cell, and they have different types performing many different functions. The prominent 3 plastids are the chloroplast, which you already know that chloroplast is a structure which contains chlorophyll pigment, and that is why it is green in color. So it is a chlorophyll containing cell organelle, and it helps in photosynthesis. Another type of plastid is which is not green in color and does not contain chlorophyll, but contains different colors, like it contains yellow pigments like xanthophyll, red pigments like carotenoids. So chromoplast are the plastids which have different colors, yellow, red, orange type of pigments. They are mostly found in flowers and fruits. The third type of plastid are the leucoplast, which does not contain any pigment or any other pigments or the colors. The main function of leucoplast is to store the starch. Okay, so chloroplast function is photosynthesis because it contains green chlorophyll pigment. Chromoplast function is to impart colors to flowers and fruit, and apart from green they contain red, orange and yellow pigments. Leucoplast on the other hand are the plastids which do not contain any pigment and helps in storing the starch, right? So now comes the next one. Let's study in chloroplast in detail. Right now, chloroplast, like the mitochondria, is again a double membrane structure. Mitochondria also had a double membrane. Plastids also have 2 membranes, outer membrane and inner membrane. The space between the 2 membrane is known as intermembrane space. Inside the inner membrane there is a gel-like substance, fluid-like substance, which is known as stroma. Over the stroma, filled inside or embedded inside it, are present some coins-like structure, stack of coins-like structure. So one of those coins is known as thylakoid, and a group of thylakoid is known as granum. Okay, so it is the thylakoid and the granum where the actual photosynthesis takes place. All the chlorophyll pigments are present inside the thylakoid, and it is the thylakoid which helps in photosynthesis. These granum are joined by a lamella, okay, where the dark phase of the photosynthesis takes place. Now if we see about Plastids in plants, can you identify them? Let's see. Flower is yellow color and this fruit is red color. Which type of plastid will be present? Yes, yellow, red, orange means it is a chromoplast. What will be the function of chromoplast? Chromoplast will help in flowers to attract bees for pollination, right? If we move on, in the green stem and the green leaf, which type of plastid will be found? Yes, here we will found the chloroplast because the leaf will take part in photosynthesis and it is mostly green in color. As we go down, let's see in a dry leaf, which is dying leaf, which type of plastid will be found? Well, since the plant is dying and wilting, the number of chloroplast will start decreasing. Since the number of chloroplast will start decreasing, it will not be able to perform the process of photosynthesis. It will not be able to make its own food. So hence the leaf will start wilting. If we move down to the roots, we see there is radish, carrot, like. So in the roots, which type of plastid will be found? Obviously it is inside the soil, so chloroplast cannot be here. Which type of plastid will be found? It will be leucoplast, because the only and only function of the root can be storing of starch. Clear? So now comes the last organelle, which is again prominently present only in plant cells, and that is vacuole. Vacuole is present in plant cell and they are mostly absent in animal cells, and if they are present they will be very, very small in size. What is the function of the vacuole? Vacuole are the storehouse of the cell, right? All the types of components, waste components, or it has made anything in excess, like there is some excess proteins or excess fat have been synthesized, which is not immediately in requirement. Where will the cell store? It will store inside the vacuole, right? Apart from this, vacuole also helps in maintaining the osmoregularity of the cell. You have studied about the osmosis, right? What is osmosis? Movement of particles, liquid particles across the semi-permeable membrane from high concentration to low concentration. So when the particles are continuously moving in and out of the cell, there are chances the cell may burst out, or sometimes it may are chances that it may shrink out. So vacuoles helps in maintaining that tonicity of the cell. So hence vacuole helps in osmoregulation also. Clear? So that was the cell organelles. With this we have concluded our chapter 6, fundamental unit of life, cell. I hope the information was useful. Go through these topics and I'll see you in the next video. Thank you.